Hello to everyone. So I'm Giorgio. I'm from Europe, in Italy. I'm a father of two children, Tango 2, Edoardo and Carlotta. Edoardo is 8 years old and Carlotta 7 years old. And now we will see a video remote from the French group. There are two doctors that are working in the last years on Tango 2. In particular, I'm talking about Pascale de Lonlay. She's a doctor from Necker Hospital in Paris, and she's a pediatrician, and she's expert on metabolic diseases. In particular, she's an expert on energetic metabolism and molecular biology and biochemistry and genetic and cell biology. Sorry for my English. I don't use these kind of words normally. Okay, and then Dr. Hortense de Calbiac. She's a postdoc. She works with Pascale de Lonlay, but also she has worked for a long time with Edor Kabashi. Edor Kabashi is another doctor. He was working in other research group. We have seen before with Mike's presentation that in French there were two different research activities, and she was studying at Sorbonne University, and now she's working with Pascale de Lonlay. Unfortunately, they cannot be here. 3 years ago, there were representatives here in USA, but this time they cannot attend. So now we can take a look on the video, and of course, if there are any questions, we can also raise a question and send by mail to them and get the feedback if we want to have more information on the research that they are running. Thank you very much.
So good morning, everybody. With Hortense de Calbiac, who is a researcher, and Pascale de Lonlay, a physician. We thank you for inviting us to speak about guidelines of Tango 2 deficiency and treatments with an animal model, zebrafish. Hortense is the specialist, and we are sorry we didn't come to Orlando because it's very far, but thank you for accepting that we speak with a video. So, as you know, with your personal experience and with your physicians and with Professor Lalani and other physicians in the Congress, you know that Tango 2 deficiency includes neurological disease, hypothyroidism, metabolic crisis, including hypoglycemia, rhabdomyolysis, encephalopathy and seizures, cardiac dysfunctions, with cardiomyopathy, arrhythmia, with long QT, with other arrhythmia like ventricular arrhythmia progressing to torsade de pointe and bruchetta paterne. So Tango 2 deficiency needs several specialists, including cardiologists and anesthetics, as we will see. And we know that we have a clinical variability between families and even between siblings. So you know the triggers of the disease for acute metabolic crisis with fasting. So the importance to administrate glucose infusion, the dehydration, so adequate hyperhydration, infection and fever. So we have to give a specific treatment for infection to limit the trigger of the decomposition. And a lot of drugs are contraindicated in Tango 2 deficiency, including drugs used in arrhythmia by cardiologists and drugs used for general anesthesia. I didn't list all the publications of Tango 2 deficiency, of course, but a few recent papers on arrhythmia and on general anesthesia. And here you have a recent paper on 27 children in cardiology units, probably, admitted for 43 cardiac crises in several centers. And as you can see, all the patients in cardiology, so it's normal, they had a cardiac problem. They all had a QT prolongation, bruchetta pattern in 26% of the cases, arrhythmia in 90% of the patients, and cardiomyopathy in 70%. And these authors described the successful treatments they used for the patients. We had one recent patient. I don't know this patient, but we had in France this patient who, in intensive care unit for rhabdomyolysis, right branch block, so arrhythmia, long QT, a coma, and the physicians used a sedation with hypnovel, sulfatanil, ketamine, and they used cordarone for the arrhythmia. And in red, the cordarone probably is not indicated in TANGO 2 deficiency, but it's a drug used by cardiologists. And he had a cardiac arrest. And another patient we reported had a surgery for orthopedic surgery, and he had anesthesia with propofol and sevoflurane, probably contraindicated. And after the surgery, he had asthenia, then rhabdomyolysis, an increase of the QT interval, and neurological deterioration. The physicians used propofol and cellular and followed by arrhythmia with ventricular fibrillation. And they used amiodarone. After that, they used L-carnitine because they suspected energetic disease. And 2 hours after injection of L-carnitine, the patient had long QT associated with Brugada pattern and 2 episodes of arrhythmia with torsade de pointe and ventricular fibrillation. So he had electric external shocks and magnesium injection, and now he is well with implanted cardiac defibrillator. So we know now that we have to share the list of drugs we can use and which are contraindicated in TANGO 2 deficiency. In this paper, nice story, the child who had a tetralogy of Fallot with TANGO 2 deficiency. So he had general anesthesia, and the author underlines that no guidelines exist, and it's a problem for the general anesthesia. And they used sevoflurane, probably contraindicated, when the patient was well after the surgery. Of course, the patient went to intensive care unit immediately after the surgery. So, of course, the quality of life is bad with the risk of metabolic decompensation with encephalopathy and arrhythmia. So we have to work all together with the list of drugs to give and to contraindicate. So the emergency protocol or emergency certificates are given to the patients. So the patients don't wait to be taken care of in emergency units. Because TANGO 2 deficiency is a new disease, we are learning from you, from the patients. So this certificate will need to be modified and shared with all specialists, cardiologists, anesthesists, etc., and different teams in the world, and of course, with the patients. We are stronger together, of course. So in these emergency protocols, the first urgent actions in case of gastroenteritis, fever are indicated. You have the priority in emergency units. This certificate explains the risk of the decompensation and list all the complications. They indicate emergency blood test with glycemia, serum electrolytes with kalemia for cardiac distress and rhabdomyolysis, creatine kinase with rhabdomyolysis, troponin, which is a marker of cardiac failure, renal function, liver function. They indicate that we have to perform a systematic electrocardiogram, plus ou moins cardiac echography, to search for long QT, Brugada, arrhythmia. In this emergency certificate, we have a systematic treatment to administrate without waiting lab results. So it's very important to have this certificate with you in all the units. So in this emergency certificate, we have the glucose infusion with sufficient quantity of glucose, depending on the age, but also depending on the cardiac function. No potassium in case of rhabdomyolysis, and we adapt the administration of electrolytes and potassium to the ionogram monitoring. no lipid, maybe no carnitine induct, and a supplementation in magnesium in case of arrhythmia because it's an anti-arrhythmic drug. We continue usual medications of the patients. We give specific treatment for infection, which are the trigger of the decompensation. And in case of rhabdomyolysis, we remove potassium, hyperhydration, but be careful with the cardiac function. In this emergency certificate, we indicate the drug to give or not, they are contraindicated in case of arrhythmia. So we need a specialized advice from a cardiologist specialized in arrhythmia. We have a risk of long QT, brugada, and arrhythmia. And our cardiologist at Necker says that the cordarone and sotalol, which are the first treatments given by cardiologists in case of arrhythmia, are contraindicated in TANGO deficiency because they induce long QT. Verapamil was used in a paper, but for our cardiologist, verapamil has no demonstrated effect in arrhythmia, long QT, brugada. In case of long QT, our cardiologist proposed beta blockers, except sotalol, because of the long QT. Beta blockers have no side effect in case of brugada, and they propose as beta blockers Corgard if there is no cardiac dysfunction, otherwise bisoprolol. They can propose Isuprel IV intravenous in acute long QT and brugada pattern, and a supplementation in magnesium. And if persistence of the arrhythmia, external electric shock, and they discuss extracorporeal membrane oxygenation, and of course the patient is in intensive care unit. So we can see that it's not the metabolician which can indicate all these drugs, but the cardiologist. And in this emergency protocol, we have the contraindicated drugs. We have a lot of drugs which prolong QT. So we have a list of drugs contraindicated in long QT, a list of drugs contraindicated in brugada, drugs containing lipids like propofol for general anesthesia, propofol which is a lipid and which is contraindicated also because of long QT. Salicylic acid, valproic acid, maybe L-carnitine, glucagon in case of hypoglycemia, some anesthetics, as we will see, fasting, and statin because they induce rhabdomyolysis, as we will see with the zebrafish model. So in this emergency protocol, we have also a paragraph for the general anesthesia with the importance of glucose infusion. Of course, scheduled surgery are in hospital with an intensive care unit and with cardiologists specialized in arrhythmia. We have some absolute contraindications like succinylcholine, which is a curare that leads to muscle contraction, which give rhabdomyolysis, propofol, as we will see, and sevoflurane, because they are depressor of the myocardium. So the anesthetics and the cardiologist propose for the induction of anesthesia ketamine and morphine drugs, plus or moins curare, rocuronium, if intubation is difficult, and maybe, in absence of long QT, propofol, maybe, but not during anesthesia. During anesthesia, they propose morphine drugs and benzodiazepine, and other drugs are in discussion now. And of course, immediately after surgery, the patient is in intensive care unit. So we have all the metabolicians have emergency certificate for each metabolic disease. So in France, each hospital have emergency certificates for their patients. And in France, we have now G2M, which is a network of all reference centers of inherited metabolic diseases that I coordinate. As now, our certificate at Necker have been corrected by all physicians in France. So it's a consensus in France for the metabolic disease. So the emergency certificates are very short, 2-page, recto verso. And for the TANGO deficiency, we didn't finish. It's in process with anesthetics and cardiologists. And of course, we have to share this protocol with you, with the international teams but mostly cardiologists and anesthetists, of course. So you can see the 2-page, but it's not finished, of TANGO 2 emergency certificate. So we have the priority in emergency units. We have here immediately the risk of arrhythmia, the risk of neurological distress and hypoglycemia rhabdomyolysis, the screening to perform immediately when the patient is in the emergency unit, the treatment with the glucose, with the magnesium in case of arrhythmia. We have all the chapter on arrhythmia with the contraindication and the drugs which are proposed. And we have the severity signs, the monitoring of the patient. And in second page, a small chapter on the physiopathology, the trigger of the decompensations, the clinical signs of deterioration, and mostly the contraindication, the drugs which are contraindicated with the list of drugs which prolong QT or which give Brugada pattern, and a chapter for general anesthesia with a lot of commentaries because it's not finished by our cardiologists for the anesthetics. So we will have to work together this emergency certificate. So now Hortense de Calbiac, who is a researcher, will present animal model to find medications.
Okay, so hi everyone, I am Hortense de Calbiac, and I'm very happy to share with you all what we are doing in the lab. So we are using a zebrafish model to study the role of TANGO 2 at whole organism scale and to test some therapeutic candidate molecules. So maybe you're not familiar with zebrafish model because it is quite recent still as a model system, even if numerous drug treatments have entered the clinic or clinical trials and have their genesis in zebrafish actually. So zebrafish display numerous advantages in our perspectives of understanding the function of our gene of interest, TANGO 2, and in the identification of potential drugs to prevent rhabdomyolysis events. So first, zebrafish is a vertebrate animal with similar biology and genome to humans, so we can translate our observations in the context of human pathology. For example, TANGO 2 gene in zebrafish display more than 90% of genome alignment with its human orthologue. So another major advantage of zebrafish is that it gives a lot of eggs which develop very quickly with, for example, a fully developed locomotor system at 2 days old. And something very important to us is that zebrafish embryos are small enough to fit into little wells and are permeable to small molecules and drug treatments at physiologically relevant dose ranges. So these features enable high throughput and high content phenotypic drugs screening. And as an example, once all the experiment plan is set up, you can measure the effects of a drug of interest in less than a week. So taking advantage of all these properties, we have developed a zebrafish model to study TANGO 2 pathology. So in zebrafish, rhabdomyolysis phenotype is well defined thanks to previously established genetic and chemical zebrafish rhabdomyolysis models. And typically it is characterized by locomotor deficiencies due to muscle structure defects. And we can use different tools that I will present and that we optimized to assess these features at different developmental stages. So TANGO 2 mutations are known to induce the loss of the protein. So to generate a zebrafish model of TANGO 2 deficiency, we want to inhibit the expression of this gene in zebrafish, or to do a knockdown of the gene. So to do so, we use a technology called morpholino oligonucleotide, or AMO, which is a little sequence of DNA that we microinject in zebrafish embryos right after the fertilization, so at zero hours post-fertilization. So the morpholino will bind to the RNA coming from TANGO2 gene, and by such a binding, it will prevent its expression, and then we will lose or decrease the corresponding protein in zebrafish. So this is how we mimic TANGO2 mutation in vivo. And then we will be able to look at the causative effects of TANGO2 depletion in vivo, which will give insights to what mechanisms we should target to restore TANGO2 loss. And then this is how we can identify some candidate molecules to screen in a therapeutic perspective. So to inhibit TANGO2 expression in zebrafish, we use two strategies with two different morpholinos. So 2 days after the fertilization, so 2 days after the injection, zebrafish embryo look like that, and we can assess their ability to move using a test called the touch evac escape response, which is a direct indicator of the muscle integrity of zebrafish. So basically what we do for this assay is to touch very lightly the tail of zebrafish with a tip, and we measure the escape to touch. Here is a video where you can see an example of a zebrafish TER test of a non-injected, so this is a control zebrafish. So from the recording of zebrafish tested with the TER assay, we can project their movements on axis and quantify locomotion parameters, such as the distance that they traveled, for example. So we have three conditions here, the control and the two different morpholinos, morpholino 1 and morpholino 2. And you can see that for morpholino 1, there is no visible change on the locomotion as compared to the control, and as quantified here for the distance. Whereas for morpholino 2, you can see a decrease of the distance that embryos can travel, which is significantly decreased as represented here compared to the control. So overall, this indicates that morpholino 2 is able to induce locomotor defects in zebrafish. So at the same stage, so still at 2 days post-fertilization, we looked at the global morphology of our zebrafish embryos, and you can see that morpholino's injections are not inducing any defects in the global anatomy, since we didn't observe any abnormalities in both morpholino 1 and morpholino 2 compared to the control. And this is just a picture of an experiment to validate the knockdown of TANGO2 at the RNA level, showing that we are actually able to inhibit the gene, the expression of TANGO2 in zebrafish. So then we wanted to look at the cellular level to see if the locomotor defects that we observed were associated with any muscular abnormalities. So we optimized the technique to look at muscle architecture by polarized light, by imaging of the zebrafish still at 2 days after the fertilization. So to correlate muscle integrity or muscle defects to the locomotor defect that we observed for morpholino 2. So this is a magnification where you can see that in the condition where we observed the locomotor defects, so using the morpholino 2, there is a global disorganization of the muscle fiber as compared to the control. So this muscle analysis, along with locomotor quantification, will serve as specific readouts for drug screening. So we are looking for drugs that can rescue this abnormal myofiber infrastructure. And then at 3 days after the fertilization, zebrafish are big enough to be detected by this machine, which is called the viewpoint system, which allows the automated tracking of zebrafish embryos. So the idea here is still to measure the locomotion traveled by each zebrafish embryo, and then potential muscular defects in an automated fashion. And what is very nice here is that you can track 96 zebrafish embryos at the same time, because we can use this kind of plate where we add 1 zebrafish per well along with the drug of interest that we simply add in zebrafish water. So this is how we do drug screening with a pretty high throughput. And now that we have optimized our readouts, we are ready, and we have actually started already to test some drug in order to rescue the phenotype using the different readouts that we identified. Then the effects of molecule able to rescue the locomotion of zebrafish will be assessed at the muscle levels. So you may know that in Pascale de Lonlay's lab, we are lucky enough to have TANGO 2 patients' primary bioblast, which are very useful model to understand the role of TANGO 2 in these cells. So the principle here is that we can translate our fundamental results obtained in patient cells to zebrafish drug screening. In practice, we will screen in zebrafish some drugs known to ameliorate the defect that we identified in primary bioblast. And now that we have characterized and optimized the readouts to target in zebrafish, we have started to test the drugs in order to rescue the phenotype. And then we isolate the effects of each drug on the locomotion of TANGO 2 zebrafish model, and we test this drug back on primary bioblast to confirm their beneficial specific effects on TANGO 2 patients' primary cells. Then, by navigating back and forth between zebrafish and primary myoblast from TANGO 2 patients, using the tools that we design in both these models, we are quite optimistic to identify therapeutic drugs to prevent rhabdomyolysis of TANGO 2 patients. But what is also nice with this model and setups is that we can also identify drugs that may trigger rhabdomyolysis in TANGO 2 patients, so that we would be able to exclude from a care protocol. So this is an example of such a test of TANGO 2 zebrafish model sensitivity to drugs known to potentially trigger rhabdomyolysis. So you may know that statin treatment can induce rhabdomyolysis events. So we tested atorvastatin, one of the best-selling statins for the treatment of hypercholesterolemia, and on our models. So we did this experiment with a tablet of ATV that we crushed and dissolved, and we have tried 3 different concentrations, 0.2, 0.5, and 1 micromolar, according to the literature. So for the lower dosage at 0.2, nothing happened in all the conditions, so it didn't do anything on the zebrafish. But at 0.5 and 1 micromolar concentrations, we obtained very interesting results, as I believe. So at the high dose, 1 micromolar, all the fish were affected, including controls. So meaning that they were not able or almost to move. But at 0.5 micromolar dose, while there was no effect on the control as shown here, we were able to see that both TANGO 2 morpholinos model were particularly sensitive to the treatment and were almost totally unable to move, as shown by the decreased distance in both these conditions where TANGO 2 is not done. So in these videos, these are the results of embryos injected with morpholino 1 upon treatment with ATV or without any treatment. And while untreated zebrafish knockdown for TANGO 2 gene display a normal behavior here, upon treatment with statin, they show rhabdomyolysis-like features. So if you remember, morpholino 1, the morpholino 1 that we use is not giving rise to any locomotor phenotype by itself, as represented here, but upon statin treatment, it actually potentiates its effects on locomotor system. So then we reconsidered morpholino 1 as a good strategy to study TANGO 2 pathology, where stress induced by statins here trigger rhabdomyolysis in vivo. So thank you very much for your attention. So this work has been done under the supervision of Pascale de Lonlay, in collaboration also with Edouard Kabashi's lab, and with the help of SFR Necker platforms. So I take the opportunity to thank everyone involved here, and especially TANGO 2 Research Foundation. And we are sorry that we are not able to answer questions in live, but if you have some, please don't hesitate to reach out or to pass them by TANGO 2 Foundation Committee. We will be very happy to answer them, of course.
Bye-bye.